Hi everyone, so right now we're going to be talking about glycerol 3-phosphate shuttle and malate aspartate shuttle. This is for transfer of reducing equivalents from cytosol into the mitochondria. Now, first of all, why are these shuttles needed? In a mitochondria membrane, if you see, this is the mitochondrial membrane, particularly inner mitochondrial membrane, this is impermeable to NADH. So you won't be able to transport this NADH from the cytosol into the mitochondrial matrix. You cannot do that because in a mitochondrial membrane is impermeable to NADH. So reducing equivalents from cytosolic NADH must be transferred indirectly to the mitochondrial EDC. So the shuttles they help with the transportation of this NADH to the mitochondrial electron transport chain. And how is that done? So first of all, glycerol 3-phosphate, it operates in brain and skeletal muscle. The first step is that from the cytosol it occurs in the cytosol where DHAP, which is dihydroxyacetone phosphate, and NADH, they're converted into glycerol 3-phosphate. So you have your dihydroxyacetone phosphate and the NADH, they come together and they get converted in the presence of this enzyme, which is cytosolic glycerol 3-phosphate dehydrogenase, and they get converted into glycerol 3-phosphate. The first step is to make glycerol 3-phosphate since this is glycerol 3-phosphate shuttle. So you made using dihydroxyacetone phosphate and NADH and glycerol 3-phosphate dehydrogenase, you made glycerol 3-phosphate. Now once you got the glycerol 3-phosphate, this is going to go to the inner. Now this can easily be transported. Now this will go into your inner mitochondrial membrane. There it's going to react with this mitochondrial glycerol 3-phosphate dehydrogenase, and for that you also need oxidized FAD, which will get converted to FADH2, and this glycerol 3-phosphate it gets converted back to DHAP, and then once it gets converted back to DHAP, that's not much of our concern. What's more of our concern is that we finally got FADH2 because of this reaction, right? FAD was sent and converted to FADH2. Now, this FADH2 is going to transfer the electrons to the coenzyme Q, which is ubiquinone, and enters the electron transport chain at the third complex because third complex is the one that receives the electrons from coenzyme Q, right? So this is the shuttle. So it's called glycerol 3-phosphate shuttle because glycerol 3-phosphate acts as a shuttle. And basically what you need are the electrons. So that's exactly what is happening here. So dihydroxyacetone phosphate in the presence of NADH gets converted to glycerol 3-phosphate. That goes in glycerol 3-phosphate dehydrogenase you have here. So this is glycerol 3-phosphate dehydrogenase 2. This is glycerol 3-phosphate dehydrogenase 1. This is cytosolic. This is mitochondrial. And that converts glycerol 3-phosphate back into DAP, but it also produces FADH2 that goes to coenzyme Q. That's it. And what's the ATP yield? You got uh, the ATP yield is each cytosolic NADH gives you 1.5 ATP. So basically 1.5 ATP was used right here. Now, another thing, key features is a very fast, irreversible, and lower ATP yield reaction. When you talk about malate aspartate shuttle, now when we look at malate aspartate shuttle, and this what happens is that you have this operates in liver, heart, and kidney. The upper one was operating in brain, skeletal muscle, and this is operating in liver, heart, and kidney. So the steps are first of all from cytosol you have oxaloacetate. That oxaloacetate combines with the NADH and it gives you malate. So oxaloacetate and NADH they combine to give you malate. This is the reversal of the TCA cycle. Now this happens in the presence of cytosolic malate dehydrogenase. So malate dehydrogenase, that's it, easy. And yep, malate dehydrogenase with cytosolic. Now this malate is transported across the inner mitochondrial membrane where the malate alpha ketoglutarate antiporters. Alpha ketoglutarate will be transported here. Antiporters, see this is alpha ketoglutarate and malate will be transported on the other side. So it's transported into the mitochondrial matrix. This was cytosol. Now we are in the mitochondrial matrix, and this malate will be converted back to oxaloacetate and it will give you NADH. So this way that NADH is going to enter the electron transfer chain at the first complex. So in this way you got your malate, giving you one NADH. So yes, this is how the entire reaction took place, and that oxaloacetate will combine with glutamate, and it will give you aspartate and alpha ketoglutarate. This alpha ketoglutarate will again transported through this antiporter, and malate is used. Malate is pumped towards the other side using the energy. So this is what happens. Now this is a comparison that you can see. The aspartate malate shuttle is slower, but its ATP yield is higher because you have NADH being produced, so that will be 2.5. Here you have FADH produced, right? In this one, you have FADH2 produced, so 1.5 ATP yield. And this is irreversible, whereas this is reversible because that aspartate malate shuttle, it is reversible. You can see that step. So yes, that's it.